This week we spoke with Professor Peter Hegemann from Humboldt University in Berlin. He has had a very long career studying photoreceptors and optobiology. He also produced some of the initial work done in Chlamydomonas in characterizing photoreceptors in that species. During our conversation with him, he talked a lot about his ups and downs of his career. He was a really wonderful person to talk to. He had a lot of really insightful comments about dealing with the fluctuations in progress and success during a long career focused on really one main topic. And he was a real joy to have a conversation with. I'm sure you'll really enjoy listening to it as well.
Yeah, so my name is Peter Hegemann. I studied chemistry in Münster, Munich, and then I did my PhD at the Max Planck Institute in Martinsried about light-activated pumps, in halorhodopsin in particular. So we purified it as a graduate student. And then the professor or the director of the institute said you can stay as an independent research group in the department, but you have to move on to a different system, which is interesting. And at the end, from a perspective 35 years later, it was a good decision because you can combine the knowledge you learned from your old research work and combine it with a new perspective. And it forces you to move on into a different system. And then we moved to Chlamydomonas. And we did electrophysiology and plant physiology for 10 years or so, or 15 years. And I had my research group there at the Max Planck Institute after a short postdoc in Syracuse, upstate New York. So funding for this research was very difficult because people were not interested in any electrophysiology on green algae. So the research project seemed to be very exotic and not going anywhere. And then we proposed, based on the electrophysiology, that the photoreceptors or rhodopsin, which has an intrinsic ion channel. So all the senior people said, this man is crazy. He should learn what the difference between a rhodopsin and ion channel is. It has nothing to do with each other. And so the time was very difficult. And also funding was complicated. And then we proposed, or we purified rhodopsin from the ice part. It's also interesting, and proposed that this is a photoreceptors. And only two people were interested in this work, and one of them was Roger Chen from La Jolla. This is a person who worked on this GFP for a long time. And he said, if this is really a light-gated ion channel, it would be very interesting for application. So we communicated over several years, and this research was unsuccessful.
Okay.
And the reason is it was a wrong gene. So it couldn't work. And the other person, or the other group interested in this was Rainer Hedrich. Maybe one of you knows him. He's a plant electrophysiologist, and he lost his interest. So after then we discovered, my former graduate student Sunil Kataria from India, he discovered the new rhodopsins in these Japanese cDNA library that was released by a Chlamydomonas community from Japan. And then we expressed it in oocyte together with Georg Nagel and showed that this is indeed a light-gated ion channel. And again, the first person working with this was Roger Chen again, and the work was unsuccessful because the photo count was so small. So then we published this. After many difficulties we got by the publisher, as usual. If you publish something exotic, you run into all kinds of difficulties. And then we worked on a channel rhodopsin 2, and the count was 10 times larger. And then Georg Nagel and myself expressed it in HEK cells, human embryo kidney cells, which was interesting because that convinced many more scientists that this could also be used in animal systems. And then many groups started with this, and then the business, what is now called optogenetics, exploded.
Yeah.
So it tells you, if you work on something exotic, it does not necessarily mean that it's not interesting at the end.
Maybe you need time.
You need time,
Yeah.
and you have to be persistent.
Yeah.
And to convince, if you're convinced yourself that this is interesting, you have to be patient and you have to convince your colleagues. And then in 2005, I think 5 or 6 publications appeared. First of all, the first one came from Carl Deiserwold's lab in Stanford, and we collaborated with Stefan Herrlicher at Ohio. Interestingly, none of the German scientists were interested. I said, "Can you guarantee that this works?" And they say, "No, I cannot." But the young scientists from other countries, so take more risks, and they say, "Oh, this is fun. Let's try something new." And they started to work with us in Stanford and Ohio, and also Hiromu Yabo from Sendai started to work with us, and Tzu Pan was the first who expressed it in blind mice, in eyes of blind mice. And that was also spectacular, that the chlamydomonas protein was successful and recovered vision in blind mice.
Yeah.
So, and then the optogenetic business started. And so it's a quite a well-known protein now. So about 1,500 laboratories are working with general opsin.
It's very popular, hot topic.
Yeah, and then if you remember how it started, it was so difficult, and only the visionary Roger Chen always believed in this. He said, if this will work, it would be a great tool.
Yeah.
So he was definitely ahead of his time.
Do you think having his support throughout all of the challenges that you had really helped keep momentum as you kind of worked through all of the problems?
Psychologically, it might have used, but I think it is more important that the people in your surroundings. So my former supervisor, Dieter Österhirt, he supported us. He liked Strange project, and he said, keep going.
Yeah.
It will work at the end, and I think this is most interesting.
Yeah. I think that we all have experience with how difficult research and experiments can be, and that I always tell students that I mentor that 90% of the time it's not going to work, but it's that 10% of the time that is really the part that you have to hold on to in your heart so that you can continue with the failure. But I actually found during your talk, you talked a lot about the things that didn't work, or the things that you found out kind of later on weren't the right interpretation. And I found that actually very, very wonderful to hear someone talk about it, because I think most of the times when we go to see seminars, everyone talks about all of the genius, the genius that they had that came out of their work, and how it kind of underplays probably the trials that their students or they had as they went through. So I actually found it very encouraging to hear you talk about the struggles that your students or you or that your lab or your collaborators had, because I think that it really is a much more honest portrayal of the process. But how do you had mentioned that you had a graduate student trying to purify this protein that was colorless, and that was a struggle for her until she kind of progressed through. But how do you encourage your students to work on these projects that are very challenging, where they will have failures and they've got to kind of get through it?
Yeah, I think first of all you have to give the students a certain kind of security. So if the experiments don't work right away, you trust in these people that they find out the solution later on. So if you put them under pressure too much, and they lose interest, and they become nervous, and they try to look for something else, which is not so good. Also, several colleagues give to the students an easy and a difficult project, which we never do.
Okay, interesting.
What they do is, most of the time they work on the easy project. And so most of the projects my students do are quite difficult, but we find solutions in almost all cases, because if they keep their eye open, and certainly in the discussion with myself and more senior colleagues, they find on the way, in many cases, interesting things that were not expected.
Yeah.
So at the end, the graduate work looks different from what we originally planned to do.
Propose.
Yeah, so we didn't plan to look for a light-gated ion channel. We got the idea that this is a rhodopsin, and the rhodopsin in a plant cell would be interesting in one or the other direction. And then the photocounts were ultra fast, so faster than in any other system. And then we moved it into this direction slowly. And then the first publication we said they are closely connected, and then we said it's one complex. And then the third one we said this must be one protein, because there's no need for amplification and there's no need for biochemical signal transmission and things like this. So it developed over time, and I think this is important to transfer to the young researchers, that they are confident that we'll find something interesting.
I think
unusual.
your introduction is already persuasive, right?
Yeah.
If you start saying that it took you so many years and it was a hard transition, so I think every student will be convinced, yes, I can do it. I have to be persistent, as you said. So
yeah, well, I oftentimes think too about with respect to publishing and respect to security, job security or position security, being kind of more and more difficult these days, or maybe it's always just been difficult and we just as young researchers think that it's more difficult now than it was for anybody else.
yeah.
But in some cases you can either be kind of publication driven or research or results driven, or you can be driven by kind of an intellectual question,
Curiosity.
maybe, or curiosity. Yeah. And if we're with impact factor and publication listings while we're trying to apply for jobs ourselves, how to make a balance between being nervous or afraid that we aren't going to be secure because maybe our project isn't producing as many publications as we feel that it should, or we've been told that it should, versus really trying to be true to our interests, our scientific interests, as opposed to scientific result production. I don't know if that's a question.
Yeah,
as much as that.
this is I think a sad development during the past 20 years, that they count the number of publications and the impact factors. And so I was in a committee of the German Research Foundation about networks, research networks. And it's interesting if you look at other branches of science, for example, engineers. And for them, it's totally bizarre and crazy to publish so much so early. And they say, if you find something interesting, why don't you exploit it yourself instead of telling your competitor as fast as possible what needs to be done? And so their opinion is completely different from the biology. And many colleagues in the biology department, and neuroscience is even worse, they push the students to publish as fast and as high as possible. And then they are first author on a publication with 20 other authors on it. And for the number 2 to 20 or 19 or so the last one is normally the supervisor, it's very difficult to publish these papers, because a lot of their work also ends into these high impact publications, and no one is able to judge about the different contributions. So I don't like these high impact factors too much when there are 20 authors on it.
Yeah.
This is what is it? The incentive for the supervisor, and this is not so good for the students. And so if someone has a PhD project or finishes PhD and has 2 or 3 first author publications in decent journal, I judge it at least as much as someone who has a first author publication with 20 other authors on it. So, and I think if you develop something new, you cannot publish in these flashy journals right away. So it needs a development of many years until you reach that level, because you have to convince the reviewers about the quality.
Yeah.
And I remember my work, so when we published the channel adopts in one publication in Nature, they didn't accept it. They want single channel recordings, which is stupid for the photon channel, because you have to go to pH 0 or something.
Yeah.
But the editor insisted that this is necessary. So totally stupid from these days.
Mm-hmm.
And it's experience of many. For example, the work from Zuo Pan, he was the first who submitted an optogenetics publication. It was turned down and rejected twice. And then the next person came, which was Boton Roska from Basel. So he repeated it and improved the expression level to 1 cell types in the mouse eye. So he got it into science right away. But the pioneering work was not published in such a high impact journal. So life is unfair.
Yeah.
And then application comes into business. So to publish our Chlamydomonas work was more difficult, but then we published a number of papers with Carl Dijsselrood about mutants.
Mm-hmm.
So normally you cannot publish 1 or 2 mutants in a Nature paper, but if it's applicable to neuroscience,
then it's there.
They got it into Nature Neuroscience.
Yeah.
Yeah, there seems to be a kind of contradictory between being able to prove that you can do the things that you do well and everything also having to be novel. So how do you do novel work if you have to show that you've done it before? And
and application always puts a bias on the publication quality. So if something is applicable, it doesn't mean that the quality is enormous or the novelty is great, but application moves it up one step further.
Yeah.
And if you compare plant science researchers, the chance that they publish in high impact journals is very low compared to neuroscientists, because everyone thinks this is a new breakthrough for understanding the brain, which is not the case.
Yeah.
So the progress in the neuroscience is relatively slow. Although they publish in these high flashy journals.
Exactly.
Yeah.
This is what I was saying. Yeah, they publish. Yeah.
We talk a lot about being cell biologists and being interested in fundamental biological questions, and always getting the question, oh, but how does this apply to like crop science? How does this apply to food safety? And you say, well, it doesn't really, but when you read papers about cancer biology, how much of it is directly applicable to cancer treatment and cancer cures? None. But we use these cell systems as models to understand the cell biology behind it, but we're kind of pressured to apply it somehow, which has to be somewhat interpretive, because there really isn't a direct application to a lot of the important work that we're trying to do.
I think this is one of the biggest handicaps for EU network grants. They always want to have companies. This is in most cases much too early. So big companies are not interested because they say, if it's applicable, we take it, whether we get a grant or not.
Yeah.
But to connect it to companies is restricting these grants to application much too early. So
you mentioned kind of starting out your career in Chlamydomonas, but now working a lot with neuroscientists, and we've talked with a couple other seminar speakers who have been able to take their work and collaborate with very different fields. How do you find fitting your interests in with the cultures of different fields and research fields?
I think the most important requirement for good collaboration is a good communication with the person you're collaborating with. So no matter what field it is. So we have good and bad collaborations, and most are very interesting. And it depends on these individuals. For example, we nicely collaborate with Carl Dijsselrood. So he's one of the most influential neuroscientists in the United States now, although he's pretty young. But he studied molecular biology before he started his medical school. So he has a much better understanding of the molecular background than most other neuroscientists. And therefore it's easier with him to collaborate than with others. And he is also relatively patient. So when we start a project, he is investing time and postdocs and students. Sometimes it's difficult for us. So when we start a project with one graduate student, he adds 2 postdocs onto that project. And maybe there's a chance that he's running away. But altogether, this is a very fruitful collaboration. On the other hand, we also work with John Kennis from Amsterdam. He's a femtosecond spectroscopist. It's a quite different world. And they never talk to each other because they're so far away.
Yeah.
And then we came back to Chlamydomonas field some time ago. So it's highly appreciated now that we established this reverse genetic system.
Yeah.
Which has nothing to do with neuroscience.
Keep it safe. Away from it.
Take a breather.
Yeah, but it was also a long story. So it was not ultra competitive because people don't want to invest in methods if this takes too much time.
Yeah.
And then you are relatively safe. You can continue
Free
your
to
work,
do-
free
yeah.
to do whatever you like.
Yeah.
And then you come back when the problem is solved.
Yeah.
Yeah, I'm really interested in what you said about how kind of biologists have their own way of thinking about when we publish, how we publish, and engineers have their ideas of when they publish, how they publish, and we've talked before with some other faculty about people who collaborate with mathematicians who publish all the time or are very interested in pre-printing, and they think, or computer science, just they have a very different culture, and I always find that fascinating because I always think that everyone publishes the same way.
No, okay. And the mathematicians, it's interesting the tradition there because they have no reviewing system.
Oh, right. Yeah. That's it.
So
Yeah.
to review a high impact mathematics paper, it takes months.
Yeah.
Because you have to go through all the details
Yeah,
and to
yeah,
understand it, and so they didn't find any reviewers anymore. And sometimes this is so specialized that only 2 or 3 people in the world which know you already are reading it, and so sometimes they ask the question: Is it even worth to publish? Because 2 or 3 people that are interested know it already. And here in the biology, you want to reach a huge audience, and this is in particular a problem with the neurosciences. So if you go to a neuroscience meeting, there are 40,000 people.
Wow.
Which is completely different for the biology community.
Yeah.
It's very small.
Smaller.
So if you go to a World Conference, 150 people or so.
Yeah. Diverse though, but
This is quite diverse, and you know them for a long time.
Yeah.
So for example, John Christie and myself, we know each other for a long time, and so this is a different relation.
Yeah.
And but it has also the advantage if you collaborate with people over a long time. You don't need contracts that you need for a company.
Yeah.
So if they say we do it, and you ask them half a year ago, how's the progress, and then you trust him. He did his best that he can. Yeah. And so it's working on a totally different level. And, you have to build up your confidence by exchanging students, and they like to work in other laboratories, and so it's nice. So
Yeah, I agree. I think collaborations are useful because you can share tools, you can share reagents, you can kind of have a bigger hive mind, maybe.
Human power.
Yeah,
as you
human
said,
power,
that
yeah.
you have a student and then you have somebody bringing on 2
Other
more
things.
postdocs to
Yeah.
help. So it's,
Yeah.
Kind of share the load.
Yeah.
But certainly, especially I think if you're talking about cross-field collaborations, like you said, communication becomes kind of the linchpin that either pulls it all together or allows it to fall apart, maybe.
yeah, and sometimes the pressure on collaboration that comes from the university is too strong. So they expect that you collaborate with anybody in the department, which normally doesn't work, because I would like to collaborate with people. They like my research, and not to be forced to collaborate with the person next door, which I have no overlap with. So I think this is still the great advantage of universities compared to other systems that we are relatively free. In Germany, we have the difficulties that we have different levels of research. So this is a multi-class system.
Okay.
So we have the highly funded, extraordinary institutions like Max Planck or Helmholtz. They get huge amount of money, and the politicians want the universities to be compared with, or the output should be compared with the best universities in the world. For example, in Europe, there are Cambridge and Oxford and the ETH in Zurich. But the system of these universities is completely different. So much better funded than the average German university. So most of the money goes to extraordinary research institutions. So, or from the other perspective, these groups are much better funded than we are. So Max Planck is free, but Helmholtz has to do research that are designed or envisioned by the politicians.
Okay.
So this is different.
I think about this, the difference between having kind of equitable comparison of what is required of the research project versus what is able to be done. Just because we have PCR doesn't mean that it's necessary in every single case.
It's unrealistic, and also if you have a small grant with one graduate student, you cannot do everything. You have to publish after 3 years because he or she needs his publication for the PhD.
To graduate.
And you cannot request that it expresses a protein in this and this and this system. And why don't you use an insect cell system? It's not so easy to establish and costs a lot of money. And then they request X-ray structure would be nice. Why don't you do this? That's stupid.
Do you think that you can respond to reviewers saying this is graduate student work, they need a publication, is this really necessary for this publication, or do you go to a different journal that maybe is more specialized or less
sometimes
high impact?
We do. We cannot push everything in this flashy journal. So sometimes we try, and in other cases, for example, our photoactivated cyclase was not taken by Science. So we published in the GABC. It's one of our most cited publications because it's widely applied. So it's a good publication. And if you remember the GFP business, the most cited paper in this business is a JBC publication from Roger Chen. It was 18,000 times cited. This is a JBC because the others didn't want it. And then when the field becomes famous, then you can publish all kinds of in excellent journals. So then they know you need to publish this because it gives high citations and things like this.
The traffic and
This is not a good politic, but I think in every place there are human beings behind it, and this is also the case in these journals. So sometimes they have pretty young editors, not far away from their postdoc time, and they should judge whether something is interesting or not, which is different. And what I'm telling my colleagues over years, to review something interesting is much more difficult than to review a streamlined paper. So if they make all kinds of mutations on channel adoption, it takes me half an hour to review this. But if someone comes with something completely new, it takes me the whole week. You have to read something because you are not so familiar, and I just read a paper about upconversion of an infrared light to blue light and to application in optogenetics. It was very complicated to understand, and to judge whether it's good or not, as long as it doesn't work, is difficult. And therefore, to judge about novel science is not easy to predict. And I like to cite a classic story. So maybe you are also interested in this. So when Columbus wanted 3 ships to go to India, to find a way to India, then he went to the king, and the king said, no, it's too far. And the king asked all the astronomers, and the astronomers said, it's too far. So what we calculated, the way to India is too far. And he did it twice, and it was rejected both times. And the third time he went to the princess.
Mm-hmm.
And the princess says, if there's a person who is inquiring 3 times, we should give him the money. And then the princess was punished by the father, because he said, all people will be dead, because the journey is much longer than this naive sailor is thinking. And then he discovered America. Was it a good decision?
That's a tricky question.
I think it was a good decision.
Yeah,
I'm biased.
coming from America, yeah.
Yeah, so it only should make clear that
Well, that's a nice story.
if someone wants to do something crazy, it's difficult.
Well, and it kind of speaks to perspective too, doesn't it? I mean, it's maybe very difficult in the moment to look at something that is novel and doesn't necessarily have an application that we're used to seeing, to be able to have enough foresight to say, oh, this is going to make such a big difference in like 5 years. So I think, we hear those kind of paradigm shift stories all the time. It's like, oh, little did we know that.
But this is how science has evolved, right?
But I feel like it's again this kind of push and pull between dogma and wanting to be on the frontier. That we have dogma that we follow, that we trust, that we build our foundations on, and anytime someone says, maybe there's something different about this, we don't want to put too much into it until we finally accept it and say, okay, good, this is fine. Now this is our new dogma, and we hold on to that.
I think it's the most difficult part in your science career, to find a new research project. It's not funding or something. It must be interesting enough to keep you going for 5 or 10 years.
Yeah.
Without knowing the outcome.
Yeah.
So did you have that sense when you started your own group that staying with optobiology or staying with the rhodopsin or that this was something that you could build a career on? Or did you see a trajectory, or were you just like, this is what I'm interested in now, hopefully I'll be able to do it for a while?
Yeah, we are not so naive. So after 5 years, first we purified the wrong gene, and then I was a professor in Regensburg already. So we decided to build a farmhouse there, because we said there's no chance to move any.
Yeah.
Plan B.
Yeah, so we are pretty safe. I was an associate professor at the Regensburg University, so I said, the most difficult part is over. But after this devastating finding, we didn't expect a fast recovery. So, but we were still interested in algae research, and there were many different phenomena. So we kept going, and then we found another rhodopsin, which was more interesting. So it happens. It goes up and down all the time. And then it depends also on the family situation, on your partner, and then your partner is moving to a different place, and you are not progressing, and then you become frustrated. This is pretty normal.
Yeah. That's true. Yeah, we are discussing a lot about it, and with our speakers as well. This is a big part of our podcast series.
Kind of finding. Well, I always say I don't like the term balance between life and work, because I don't think that you can actually balance your life and your work. I think it's all your life. I think you have to just do it, I guess. But when you start throwing other people into the mix, your friends, your family, whatever, it's like more than just your life. The balance is between your life and someone else's life, maybe more than your own work and your home life. That's just what it is. Maybe. But
It's hard though.
But it's hard.
And you also have to survive the complaints from your partner, and you work until 10 at night, and so you are in a bad mood because the experiment didn't work, and then you come home and get complaints again.
But then you find this amazing mutation that turns the rhodopsin into chloride channel now, and you have a champagne.
And someone to drink it with.
So it goes up and down. I think this is normal. And also, we shouldn't focus on careers of our graduate students too much in the sense that we want to push them into science. So there are many different options. So at the end of the PhD, if it's, let's say, from good or moderate level, then we should also convince them to do something else. And after a short time of a couple of years, they're sometimes happier than if they would have been if they would have stayed in science. Some people are very successful managers or do other things.
It can be intimidating to think about what else you would ever do.
Maybe that's the problem, because if
this.
you have people like Peter that can give alternatives or pathways.
Try something else.
Yeah, it's good to have people that are already in academia, giving you some alternative ideas.
Or acknowledging that alternatives are appropriate. That they're acceptable. Because I think too, we definitely look to our mentors for their acceptance a lot of times, and you can be afraid of hurting their feelings. I don't know, making them disappointed. It's a complicated relationship.
This is what I meant about guilty thought.
So if you would be a university professor, what would you change in the system? What you don't like?
I think in general, I don't like what we discussed before, that there is this imbalance of what it is asked in practice in order to find a job. Let's say you want many publications or high-cited papers, although the focus on the university should be to build up on knowledge, to be free to investigate your curiosity, be free to be curious. So there is this imbalance between production and having the room to think about ideas that might be productive at the end, but you need to have this free space, So I don't know how I would change that, because that means the whole system has to change, not only the university, it's also the money that they come from funding bodies, from the government. So it's a more global change, but I don't know. I know what I don't like, so that is a good start.
It may be the only place to start. I think I would change the way we talk about expectations for students and career paths, or the expectations for degree programs. I think a lot of the things that I struggled with as a PhD student, and maybe even now as a postdoc, is understanding the expectations of what I'm supposed to be doing and what I'm expected to do later. I think that I feel expected to stay in academia because I've been in academia the whole time, but I don't necessarily know what is expected of academics all the time either. Are you supposed to focus on teaching or training or your own research? Are you supposed to stay, or is it just funding? Is it finding collaboration?
Altogether, probably. This is what
it is altogether, but that's unrealistic. I mean, you can't necessarily spend all of your time on all of them. So I think...
how do you find the time to mentor your students, to apply for grants, to build up network, to go to other countries to give a seminar? How do you manage to this whole business of being a professor?
I think at the moment it's not so difficult anymore because I'm funded by a foundation, so I don't have to teach anymore.
Okay.
Which is a great relief, although I like teaching. To interact with the students is necessary also to acquire good students.
Yeah.
But no, I'm more flexible than I have been before.
Mm-hmm.
But on the other hand, you need students you can rely on, so that you don't have to communicate with them every day.
Mm-hmm.
So if the students are independent and collaborating well, they learn from the other students and postdocs
Mm-hmm.
quite a lot, and then we have a general discussion,
Mm-hmm.
and so that works. And also, I sent them to a lab to do, let's say, Raman spectroscopy or ultrafast spectroscopy. I need confidence that they get along in the new lab pretty well. So I also depend on the colleagues.
Mm-hmm.
That they not complain about all the difficulties they have in the other lab.
Yeah.
So in most cases, it works pretty smoothly.
Mm.
So delegation.
Delegation, yeah, it's important. Delegation. So only if there's collaboration in the lab and in the group and in the network with other groups works smoothly, then you can do other things.
Yeah.
And sometimes you don't feel so well if you have too many other administrative things. So then sometimes also co-workers complain that you didn't find time to go over the manuscript and send back the corrections fast enough.
Mm-hmm.
Also, this is normal.
Yeah, it's just normal, I guess. You don't have enough time then. That's
You never have
maybe
enough time. You have a priority. Yeah, and then you get 20 grants from the German Research Foundation, and then the president asks, "Can you work for us now? It's time to work in a large committee." It takes a lot of time away. In addition,
Maybe having confidence to say, I know how to do my job and I know how to prioritize my time is where you have to kind of, that's when you come into yourself as
which
a
is more important
professional.
than to react to expectations from others. You have to organize your own time and say, "This is my first priority," which is normally the family if you're a postdoc or have children. And then it's also interesting to observe that young mothers are extremely well organized. So they cannot afford to have long talks on the floor, on the hallway,
Yeah.
and they have to organize their lives even better than others.
I think, mothers who are also working clearly have to do that. And they're a good example of, I think, people who have figured out how to optimize their time because they already have this one component of their life that is already required of them. So anything additional, they kind of have to really maximize.
I think we have to wrap up.
Okay.
Do you use social media at all? Do you use Twitter or not?
No.
Okay.
But
Excellent.
most of my students are doing this, or
Good.
many at least.
Mm-hmm.
Right.
So what people expect that we are interested in making connections. It's just the opposite. So if you
Okay.
get 100 emails every day, you have to find ways
Yep.
to get rid of them.
Yeah.
All kinds of
Spam.
commercials, spam, and so on. And
Yeah.
we have to find a very small note how to unsubscribe.
Right.
Yeah. Yeah.
And because otherwise you get it every day again.
Yeah.
And this is also important. But for young scientists, I think networking is more important to get connected to other laboratories, other postdocs in
Yeah.
different places of the world.
Good.
Wonderful.
Okay.
Thank
Nice.
you.
So
Thank you so much for your
Yeah.
time.
Appreciate it.
good luck for your research and for your
Thanks.
private life. And
Thank you.
find the right balance.
You can see a list of all of our invited speakers on the Institute of Molecular Cell and Systems Biology website at the University of Glasgow, where you can also find links to our previous podcasts. You can also follow the Institute on Twitter, as well as on Facebook. You can follow me, Emily Larson, at ER_Larson_PhD, and you can always follow Maria at M_Papanatsu as well. as we move forward with our podcast, we'd really like to hear more from our listeners so that we can keep the conversation going. So if you have any comments or suggestions for how we might be able to improve our podcast or what else we might be able to include, we'd love to hear from you either on Twitter or on Facebook. So please get in touch.